dephosphorylate by EIF2 phosphatase. And this kinase, will phosphorylate it, and the phosphatase dephosphorylate. This guy, the dephosphorylated form, is active in the initiation of translation. This guy here is inactive in the initiation of translation. So cells can regulate by the level of this kinase here whether or not translation is initiated. And in different cells, there are different types of kinases that regulate that. For instance, in red blood cells, there's a kinase here that phosphorylates this, and this kinase is dependent on the presence of iron in the cell. So when there's iron in the cell, it binds to, the phosphorylation is dependent on the absence of iron in the cell. So this kinase is iron dependent. If there's iron in the red blood cell, then the iron binds to this kinase and activates it and phosphorylates it. I always have to think about this one. This guy is inactive in the presence of iron. In the absence of iron, he's active. Okay? So if there's no iron in the cell, it doesn't bind to this kinase, it activates the kinase. The kinase phosphorylates the IF2, it's phosphorylated, and it shuts down protein synthesis. Right? Well, that makes sense because in a red blood cell, 95% of the protein that that cell is making, once it doesn't have nucleus, is globin. And you can't make hemoglobin unless you have iron there. So if there's no iron in the cell, why waste your time making globin? All your energy, all your amino acids, and everything to make globin. Because the half-life of red blood cell is small, it's 30 days or so. And it's dependent on them, all the translation that goes on. If there's no translation going on, that extends the half-life of the cell. Right? So in the absence of iron, this guy is activated, phosphorylates this, shuts down protein synthesis, you don't make globin, because it would be useless because there's no iron. When the cell has iron there, right, the iron binds to this kinase and activates it. The phosphatases are working all the time. This is really a cycle. So in the absence of iron, this guy's phosphorylating, the phosphatases take it right off, but this guy's putting it right back on. Right? So it's a cycle. As soon as this guy's inactivated, because he's bound to iron, this phosphatase now takes that phosphate out, activates this, initiates translation. This is an active form of translation. It initiates it, and then there's plenty of iron there, so the cell makes globin and makes hemoglobin. Don't follow that? So cells regulate with these by messing around with these initiation factors that are found that are needed to initiate protein synthesis in the cell. You either turn it on or off. All this slide shows you here is that the reason that this guy actually stops protein synthesis is that when EIF2 is phosphorylated, it binds to another protein that's called EIF2B. It forms this complex here. And this complex here essentially takes this phosphorylated EIF and pulls it, it complexes it, pulls it out of action, right? It's no longer able to do that. There's a limiting amount. The amount of EIF2 is limited in the cell. So much translation is going on. It's in that initiation step. We need a lot of it all the time. When it's complex to this guy here, EIF2B is not available for translation. It's turned off. And I think if you read that, it'll tell you that. Okay, regulation. So what happens to these proteins after they're synthesized in cells? Well, we all know that proteins, most proteins that are made are, as they come off, in fact, all proteins, all basic proteins that are made by ribosomes are pretty much useless. They have to go through a very intricate folding process. And that folding process is usually a difficult process. You usually need a lot of helpers. Now, that's what we're talking about, the helpers that fold these proteins. But there are 3 or 4 primary steps that are needed for protein synthesis. The first one is just folding. It's based simply on the primary structure of a protein. There are very few proteins. There are some proteins that, when you fold them like that, they're able to fold on their own and form a functional protein. fold just due to the primary structure into a functional protein. But there's not a lot of them. By far they're in the minority. Most proteins do need help. After the primary structure, there are several other things that are involved in folding. One is covalent binding of cofactors, vitamins, right? So the binding of cofactors or vitamins, which that little red dot is, help the folding process. One of the functions of these is to fold properly. A lot of times when we talk about cofactors, they're metal carriers, like an iron sulfur cluster. But iron sulfur clusters are also important for folding. They determine how protein folds to build an atom. So they're important. Second thing is covalent modification of that protein. Proteins, individual amino acids can be modified in many ways. If you're at UGA and you don't know that they can be glycosylated added sugars, don't tell the people over at CCRC. So they can be glycosylated, they can be sulfated, they can be phosphorylated, they can be methylated, all kinds of modifications to proteins. And those modifications primarily determine folding. The addition of a sugar, as you'll see later, is important in the folding of most membrane-bound proteins. It helps the folding process. And then finally, the last step after all of these things is the formation of a protein molecule, which is built by the bringing together of more than one protein, or more than one peptide. So that these mature functional proteins are often polypeptide. They have more than one protein that's associated. Often these covalent modifications are needed to recruit the other proteins that are needed at an enzyme, a multi-subunit enzyme. So all of these steps go on in folding, and most of them, because they are so complex, require chaperones. Hydrogenases that bacterial cells make, nitrogenases that bacterial cells make. If you look at the operon, what's an operon? Cluster of genes regulated by one operator, right? One promoter, one operator regulating the expression of a cluster of genes. Hydrogenase and nitrogenase occur in operons that have about 20 genes. So one promoter is regulating 20 genes. If you look at them, there are 2 or 3 maybe of those genes, 1 or 2, that are subunits of the actual protein. Most of the others in there are involved in this folding process, putting cofactors, iron sulfur clusters in the right place, irons in the right place to direct the phosphorylation or modification of the protein. So there's a lot of them involved. Okay, that's all I'm going to say directly about proteins. I'll talk a little bit about folding, when it occurs, where it occurs. Most proteins, as they're being translated, we'll see later. This cartoon is kind of funny, but it's actually fairly accurate. There's a hole in the large ribosomal subunit through which the nascent peptide comes out. It takes about 25, so remember translation is occurring at this interface here. It takes about 25 amino acids or so, 20 to 25, before that protein starts popping through the hole in the large subunit. But as soon as it does, that chain there, that polypeptide chain begins to fold. So proteins begin to fold as they come on. And usually, as the folding occurs, they often fold in domains. In other words, here's the amino terminal end of the protein. It folds, all the amino acids interact, perhaps even a cofactor is added. And that region is folded, and then as protein synthesis occurs, there's a second region that comes out and folds on its own. So proteins fold in domains, and as we said before, those domains are often functional. Each domain has a slightly different, can have different functions. Okay. When proteins begin to fold, they go through many different phases. This is just to show you 2 things. First of all, that as both proteins begin to fold, they go through this stage here, which we call the molten polypeptide stage. And this is a stage where proteins, you can see they're beginning to interact, but they're not tightly interacting in there, and they're not correctly interacted. They go from this molten sort of a stage to their correctly folded stage where the proteins are together. And what this slide indicates is that there are a few proteins that can just go directly, as I said, to fold. But sort of another from a chaperone. And these proteins are, these chaperones help to fold the proteins as well as to screen the folding process to be sure they're folded correctly. And during this time, proteins go through a state that we call breathing. So that what that essentially, by saying breathing, what that means is that the bonds that are forming here between different amino acids, bonds, disulfide bonds, different bonds, are able to essentially let loose, come back together. So proteins essentially experiment on folding. They breathe and lose interactions and then come back and fold. And that process continues until the binding energy that's required to hold them together in this form is that minimized. Structural people call it minimization, this process, this breathing thing we call minimization, where the energy for binding is minimized. But the idea is that during this process here, right, this is not a necessarily, it's a dynamic process. It changes all the time. Proteins experiment, they let loose, they come back together. And in order to do that, to refold this stuff, chaperones are generally needed, and I'm going to tell you about that. These screening molecules also are